Okay, before I get into these slides, once again, what we're going to go through today is our taxonomy and systematics PowerPoint. Both of those are available on D2L. I've grouped them together and added a few last-minute pictures like I tend to do, and we're going to click through these slides. I had the reminder that Quiz 1 goes away at midnight tonight, and Quiz 2 is now available on D2L. The first half of this PowerPoint is all about taxonomy, which we will see is all about the naming of organisms, classifying organisms. It literally translates into arrangement method, and it was originally developed by Carolus Linnaeus. We will consider him the father of taxonomy. And there are some characteristics that we need to know when we're giving names to organisms. The first is that the names are given a two-part name, this binomial nomenclature. And you can see that here. The two-part name is genus and species. The first part is the genus, the second part is the species name. And this is going to be helpful for multiple reasons, as opposed to just considering the common name of this organism, which is a leopard. If we think about leopards themselves, they're part of a genus of cats called Panthera, and there are multiple species. I added this picture just to show you a variety of different leopard species. You can see that they all have the same genus name, but they have separate species names, kind of indicating a difference between the two. And I liked this picture because it also shows the different geographic regions that each of those different species of leopard evolved in, showing us that allopatric speciation. A couple of handouts that are fresh for today. One of those is an updated class schedule because the one that I originally gave out had a day on there that we didn't actually meet, so I had to adjust those numbers. The second thing that you have there is a phylogenetic tree of all of the eukaryotic organisms that looks complex right now, but by the end of this PowerPoint, when we go through the systematics part, we should be able to make sense of those branch points and evolutionary relationships between those groups. And just to go a couple of steps back here, in the first part, taxonomy, we're just going over how organisms are named. And we mentioned there's the two-part naming. Let me try that again. We had mentioned that each name has two parts to it, this binomial nomenclature. And the second thing that we'll mention in the second half of the taxonomy PowerPoint is this idea of nesting. We'll see this hierarchy of smaller groups nested inside larger groups. You kind of saw that with this slide. Smaller groups, being the species, are nested inside larger groups, the genus. And there are even larger groups still from the genus, these groups that we will call families. And we have more and more inclusive groups as we zoom out. The most inclusive and broad groups are going to be the domains. We will recognize 3 different domains. But I just talked about the names, that there is this two-part name, the genus and species name. And we will see that organisms that are members of the same genus are going to be more closely related to one another than organisms that have different genus names. The two-part name, I said the first part is the genus, and the second part of the name is the specific epithet, which is something unique about the organism. It can sometimes be descriptive of the organism, it may be descriptive of the region that you find the organism in, or it may be descriptive of the person that found the organism and named it in the first place. If we can adopt these scientific names, it will help alleviate confusion when you're talking about organisms in the wild. Like, this is a yucca plant that you might see if you go for walks in the hill country, and there are several different species of yucca. And if you go just by the common name of this organism, which there are several common names, it could be confusing as to which plant you're talking about. Bear grass or Adam's needle or weak leaf yucca are all common names for this plant. But the scientific name is Yucca filamentosa. You can see the genus and species name. There's a couple of other things that are notable, that genus and species name is italicized. The second part there, the specific epithet, is descriptive of the organism. In this case, you can see these tiny little filaments that are peeling away from the edges of the leaves. That descriptive feature is where this species gets its name. There are other ones. This is maybe one of my favorite ones. I had another picture right here. I don't know why that didn't pop up. But these are snakes that you see in this region that go by multiple common names. People will see these snakes in a cornfield and call them a corn snake, or they'll see them eating a rat and they'll call them a rat snake. It's the same snake. The genus is Elaph, and this species is Gutata. So Elaph gutata is the scientific name, even though this has multiple common names. They have a wide variety of scale patterns, but it's all the same species. This is, once again, kind of showing us a group of organisms that if we go by the common name, you might not immediately tell which organisms are more closely related to who. The lion, the jaguar, and the leopard, and then this snow leopard down here, they all sound relatively different, but these two leopards sound like they might be relatively similar. They're both leopards, but when you go into the scientific naming of this, you can see that it tries to show something about evolutionary relationships. We can see that the lion and the jaguar and the leopard are more closely related to one another than either of them are to the snow leopards. The snow leopards have their own genus, Uncia. So genus and species would be Uncia. Uncia. The other ones are Panthera leo or Panthera onca or Panthera pardus. Again, if they share the same genus, they're more closely related than organisms that have a separate genus. Some other housekeeping things about talking about scientific names of organisms. There are some guidelines to follow. Like if you are typing a scientific name, you'll want to make sure that it's italicized. So back here, I made sure that this was italicized. The other thing that you'll notice is that the first letter of the genus name is uppercase, and everything else is lowercase. Even the species name is all lowercase. If we're handwriting this, if I'm putting something on the board, I can't italicize it on the board. But if we're handwriting it, we want to make sure that the genus and species name are always underlined. Again, simple guidelines. We will get used to seeing these as we go through. Okay, I just said that. So let's look at some examples of genus and species names. Here's a example of genus and species. Panthalops hodgsonii is the genus and species name of these Tibetan antelopes. They were originally discovered by a person, Brian Hodgson, and he decided to name these organisms after himself. So Panthalops is the genus, but this particular species that he discovered, he named after himself, Hodgsonii. So sometimes species names, like we said, are descriptive not of the organism or the area that it comes from, but of the person that described it. But there was one of the spiders or something that was discovered in the last few years that somebody named after Stephen Colbert or something like that. If you discover the species, you can kind of name it whatever you want to name it. Another example would be these possums, the Didelphis virginiana. This species comes from the fact that it was originally documented in Virginia. So it is sometimes descriptive of the region that it comes from. The next example that we see here is kind of a mouthful, Myrmacophaga tridactyla. Again, weird to say, but it translates into eater of ants with 3 fingers. And really those fingers are 3 big claws that it uses to till up the ground. And as it rips up that ground, it stirs up ants' nests and it licks up those soft-bodied insects. Remember, we will learn that these are highly derived mammals. They have become so specialized that they've lost their teeth and they just have a long, sticky tongue. The second part here is all about the hierarchy. And we saw this in a couple of slides earlier. The most inclusive and broad group is going to be domains. We will recognize 3 domains. And historically, there were, underneath the domains, 5 kingdoms. 2 of those have since been removed because as molecular tools have come about in the last couple of decades, it has allowed us to kind of rewrite this evolutionary tree. Kingdom Monera, which used to be a group that all prokaryotic cells were grouped into, has now been gone away with. It's been split into 2 domains. And then this Kingdom Protista has been grouped in with the plants, the fungus, and the animals because they're all made out of eukaryotic cells. Here are the three domains that I mentioned. There's this large domain that we call Eukarya, and that's the handout that I was just flashing earlier. It is something that we will see in Unit 2. We will get to know this chart in great depth. But before we dive into the details of this chart, it is helpful to be able to read these charts in the first place. So the broadest picture that we have here, the most inclusive groups are these three domains. The group of Eukarya is going to include any organism that is made out of eukaryotic cells. You can see plants, animals, and fungus are among this group. And anything else that's within this group that is not a plant, an animal, or a fungus collectively falls under the category of protists. There's a bunch of different protists. Again, you've got the chart in front of you. The other two domains that we have here are both going to be domains comprised entirely of prokaryotic cells. There are some differences between these two prokaryotic cells. None of them are going to have organelles. None of them are going to have a nucleus. They're all very tiny. But there are some key differences that we can talk about that distinguishes them from one another. Here are pictures of these things first. This domain of bacteria you can see are small cells. Even at high magnification, we can't make out internal structures. We can also not make out any internal structures when we look at zoomed in pictures of archaea. Again, these are both prokaryotic cells. But when we look at the last domain of the Eukarya group, this is once again including plants, fungus, and animals, and any organism that is made out of a eukaryotic cell that isn't a plant, an animal, or a fungus. So that broad last group there is what we call the protists. I said I would mention some differences between the bacteria clade and the archaea clade. They're both made out of prokaryotic cells, so they don't have internal organelles. They both, like we said last semester in 1406, all prokaryotic cells have their DNA in a circular loop, like a bracelet or a necklace. And we're going to see in the bacteria domain that circular loop of DNA lacks any kind of histone proteins to wrap around. Remember, your cells have little histone proteins that the DNA can spool around, and it's a way that the DNA can package itself up. The domain of bacteria lacks any of those histone proteins. It also has something in its cell wall, this substance called peptidoglycan. It's a certain type of protein only found in that bacteria domain. Then if we compare that to the other group of prokaryotic cells that we call the domain archaea, they too, since they're prokaryotic cells, have their DNA in a circular loop. However, this group has histones. It has little proteins that DNA can spool around and condense. Our cells have histones. The other thing that is notable is that the cell walls of these cells lack that substance, peptidoglycan. So if I go back to this picture right here, this was the thing that was in my head earlier that I wanted to say, and for some reason it had slipped my mind. When we look at our group, the Eukarya, we share a more recent common ancestor with the archaea group than we do with the bacteria group. So this branch point that was the common ancestor that ended up diverging into these extreme forms of bacteria or eukaryotic cells, we are more closely related to archaea than we are to those normal bacteria. Anyway. So we see the differences between bacteria and archaea. The next thing that we'll move into is the Eukarya group, which is a broad group. I gave you that chart that has all of the eukaryotic cells listed. This one is our example that we saw previously of an anteater. It's a particular type of mammal that has its own order called Xenarthrin, and I should say that X there in Xenarthrin is descriptive of the fact that if we could look at the vertebral column of these organisms, they have these transverse processes that stick out extra far, and they look like an X. So it's easy to find in the fossil record. If we find a bunch of bones, we can tell if we're looking at Xenarthrin bones because, again, they make that X. Anteaters are xenarthrans, but also all of the armadillos are xenarthrans as well. So we have that 9-banded armadillo here, and if you find armadillo bones, you will see that their vertebrae also has this X shape to it, indicating that they're also part of this xenarthran group. There are some questions in the quiz that want you to be able to distinguish the categories from organisms that you would find within those categories. The example organisms that you would find within these categories are called taxa. Here is a collection of a bunch of taxon. And the categories themselves, that is that nested hierarchy that we talked about, where the least inclusive group, the most exclusive, is the species. And then as we broaden out to domain, it becomes more and more inclusive. So we can see the species in the spotlight on this slide are black bears. Ursus americanus is the genus and species name. If we wanted to see what was more closely related, what other types of bears are there within the Ursus genus, we would see not only are there black bears, but there are polar bears and brown bears, those grizzly bears. They all share the same genus name, so that tells us they're more closely related to one another than either would be from the other bears that are part of the bear family, but not part of the same genus. This is a panda bear and that's a sloth bear. They are still part of the bear family, but they get their own genus name, indicating they're not as closely related to the other groups. Remember, the taxonomy was just going over the naming of the organism and then the particular hierarchy of species, genus, all the way to domain. The second PowerPoint that we see focuses in on trying to depict evolutionary relationships between groups, which is all systematics is about. We draw these structures called cladograms, and these cladograms are trying to depict evolutionary relationships. So part of this PowerPoint is talking about how we can read these cladograms. And a clade, just to throw that word out there again, is we can see clades. They're using different colors to show us 3 different clades. You can also see clades are nested. Smaller clades are nested inside larger clades. And then all a clade is going to be is a common ancestor and all of its descendants. So we can see this family of cats, Felidae family, is in its own self a tiny little clade of cats. But if we zoom out to look at a broader clade, there is a clade of all meat-eating mammals. That is a group called carnivores. So if we take all of the carnivores, that would be a much broader clade. Not all mammals eat meat. Some of them are plant-eating mammals. So there is still a broader clade that would include plant-eating and meat-eating mammals, kind of all the mammals. It's sometimes tricky, and I would say it's one of the great times to live in the field of biology because, like I mentioned a lot last semester, you have at your access a bunch of molecular tools that weren't around in previous decades. We can analyze DNA, we can analyze protein sequences, and this has been a much finer tooth comb when trying to analyze evolutionary relationships. It has helped rewrite a lot of these cladograms that show evolutionary relationships. So just to distinguish phylogenetics from systematics, systematics is drawing these clades that show evolutionary relationships, and phylogenetics is using the DNA tools to enhance the accuracy of those cladograms that you draw. Here's a cladogram. The process of drawing these cladograms is called cladistics. We need to be able to identify what a clade is. So we're back to this diagram, and they have, well, first off, you can see one clade. This entire picture is showing the clade of all life as we know it. There is the common ancestor of all living organisms, and a clade is going to be a common ancestor and all of its descendants. So if we grouped all of these organisms together, this would be one huge clade. And like we can see, smaller clades are nested inside of larger clades. So this is our extremely large clade. But inside that, we've got a smaller clade of just bacteria. That line represents a common ancestor, and we can see all of its descendants. Same thing with this red line. There's a common ancestor and all of its descendants, which leads to that archaea domain. And then the brown line is the line of eukaryotic cells. So there's the common ancestor of eukaryotes and all of their descendants. The tree of life shows us that it's broken up into 3 true clades. We will also see clades drawn kind of in a sideways fashion. The textbook has a lot of them this way. You can see the names of the organisms are at the tips of the branches, and out here kind of hovering away from the taxa names, we have the names of the clades. They sometimes just hover these clade names above the taxa. It's helpful because it shows you this tetrapod clade includes all of the taxa that it kind of hovers over. And then we can see nested inside this large tetrapod clade is a smaller clade that we call the amniotes. That term amniote just refers to shelled egg. So we see reptiles and mammals are going to all be amniotes, meaning that they all are capable of producing a shelled egg. You might be thinking, mammals? What are they doing in this amniote category? Primitive mammals lay a shelled egg. We are not primitive mammals. We are highly derived mammals, and we have derived another way of reproduction rather than just laying eggs. But there's still a few primitive mammals alive today that still lay eggs. We'll see some videos of them. Just like we can see amniotes or shelled eggs, all reptiles have shelled eggs. And look how we have snakes included with reptiles. We know snakes are reptiles, but if we broaden this out, snakes are included in this group of tetrapods. Tetrapods means that you have legs. Why are snakes included in this group? We will see that characteristics can be lost as species become more and more derived, as they specialize. I mentioned the anteater earlier. As it adapted to life of gumming down soft-bodied insects, it lost its teeth. We're going to see snakes derived from lizards that adapted to a burrowing lifestyle, and they lost their legs. We still include snakes as part of the tetrapod group, even though they currently no longer have legs. Their ancestors did have legs, so they remain in this tetrapod group. Okay, that's one thing. The other thing I wanted to show, which I think I've already said, this circle just represents the common ancestor of all tetrapods. So a clade, if we want to put a accurate definition on a clade, it would include a common ancestor and all of its descendants. So these little nodes or these branch points are what represent common ancestors. And if we include all of the descendants, then we've got a true clade. So just like this amniote clade, we would start right here at this branch point, and this would be the common ancestor of all of the amniotes, and it's a smaller clade nested inside that tetrapod clade. Okay, so we've seen clades. I just mentioned this, that if we switch our perspective to look at this common ancestor, then the entire clade would just be made out of reptiles and mammals. This chart shows us living reptiles and extinct reptiles. So on the tips of the branches, what we have here are a variety of different reptiles. Just because they made the list doesn't mean that they are still alive. Para-reptiles are no longer with us. Those were around during the Permian time period and then were driven extinct. Turtles are still around today. The dotted line indicates that they go really far back in geologic time, and it's not exactly clear where they branch off in the reptile tree, other than the fact that we know they're reptiles. Then there is another group of reptiles that is going to end up leading to lizards, to crocodiles, and even dinosaurs. Again, crocodiles we still have around, but the next three we don't. Pterosaurs, these were flying reptiles. Ornithischian dinosaurs are plant-eating dinosaurs, and then Saurischian dinosaurs are the meat-eating dinosaurs. We can see that birds evolved from meat-eating dinosaurs. Again, these dinosaurs are no longer with us, but we still have birds around. I'd mentioned something about last time. Birds are warm-blooded. Crocodiles are not warm-blooded, so warm-bloodedness shows up somewhere along this lineage of dinosaurs, and it's interesting to find out exactly where. Hopefully that will be illuminated in the next few years. If we keep going, plesiosaurs and ichthyosaurs, these are aquatic reptiles that ruled the deep oceans, much like sharks and whales and dolphins do today. These were the reptilian equivalent to whales and dolphins and big sharks. They also went extinct 66 million years ago with the other dinosaurs. Lipidosaurs, these are a group of lizards, basically. Tuataras are going to be very primitive lizards. Their legs are still out to the sides. Squamates are lizards whose legs are going to go more underneath their body because they can support more weight. And some of these squamates, as they move into the interior of the land, really start to diversify. Some of them lose their legs altogether and evolve into snakes. So again, snakes are still tetrapods even though they've lost their legs. The whole point that I want to make with this is the same thing that we're looking at. If we think about the common ancestor of all reptiles, and we want to draw a clade around the reptiles, we would have to have the common ancestor and all of the descendants. So that would include things like those birds. And that wasn't originally, birds weren't originally included in the reptiles, which meant that we weren't talking about a true clade. A true clade is the common ancestor and all of the descendants. If we leave one out, it's not a true clade, it's something else. And it's one of these other things. When we start drawing these cladograms, what we are trying to draw are clades. I've said that term a lot, but another word for clade is monophyletic group. So maybe put a star next to monophyletic group, because this is what we are trying to draw when we draw these cladograms. We are trying to avoid drawing the other two, paraphyletic groups or polyphyletic groups. Neither one of these are true clades. Here's what I mean. The first picture, just kind of the bird's eye view of the three, you can see a monophyletic group, same thing as a clade, is a common ancestor and all of the descendants. We didn't leave any of the descendants out from that common ancestor. We caught them all. Versus a paraphyletic group, you can see here we've got the common ancestor and we try and catch all of the descendants, but we maybe accidentally left one out. Or in the case of the birds, like we just saw, if we were trying to draw a clade around all of the reptiles, but we leave the birds out, it's not a true clade, it's a paraphyletic group. So birds, once they were included into that group, the reptiles became a true clade. There's the other thing over here that is a polyphyletic group, and this is what happens when you throw an organism into a group that doesn't share a recent common ancestor with the other organisms in that group. I'll give an example of each of these. Monophyletic groups we've seen as true clades. So that reptilian ancestor and all of the descendants, including the birds, would be a monophyletic group. Paraphyletic group would be, like we just said, trying to draw a clade around all of the reptiles, but leaving the birds out. Not a true clade. Then finally, this one is the polyphyletic group, where you've got an organism grouped in with some other organisms that don't share a recent common ancestor. I have the picture coming up, but I remember showing last semester there was a picture of a shark and then a dolphin, and then I was standing up there and I asked which one doesn't belong. And it looked like I didn't belong because I'm standing up here on legs, and the shark and the dolphin both had the flippers and the dorsal fin, and they were torpedo shaped. They were the same color. They were both aquatic predators. So this would be similar to trying to classify all of the dolphins and then throw in the sharks in there with the dolphins because they look so similar. That shark doesn't share a recent common ancestor with the other dolphins, so that wouldn't be an accurate representation of evolutionary relationships. Again, there's our shark, there's our dolphins. If we just glance at them, they look much more similar to one another than the dolphin looks to me. So this is what was kind of problematic before the last 20 or 30 years, when there were no molecular tools that were easily available. People just had to look at organisms superficially, or maybe do some dissections to see what kind of organs they had on the inside to determine these evolutionary relationships. Even this reptilian tree that I'll jump back to, archosaurs. These start to divide reptiles in terms of a 3-chambered or a 4-chambered heart. So sometimes you couldn't just look superficially, you had to go into the organs to see how the chambers of the heart start to branch to distinguish one group of reptiles from the next. Let me catch back up with my slides here. Okay, so organisms can look very similar, and that's what was so helpful about the molecular tools that allowed us to get in there and key out these differences between organisms. Superficially, this plant looks a whole lot like cactus, but they are not closely related to one another, and molecular tools have shown us that. So this is a cactus, the other one is a euphorb. It's found in the deserts of Africa. These are found in the deserts of North America. So once again, animals are very adaptable. Just by looking at their structures, you might not immediately, the true evolutionary relationships might not be immediately obvious to you. I think I pulled this up last semester when we talked about these two moles that look very similar to one another. They've both got reduced eyes, enlarged front pads. They've got wedge-shaped heads. They burrow into the ground, so they both have adaptations for tunneling, even though they don't share a recent common ancestor. Kind of like the dolphin and the shark, what they share is a similar habitat, and that habitat has shaped them in similar ways. These are what we call analogous traits. They're due to shared habitat, not due to shared recent common ancestry. We did talk about traits that were shared due to recent common ancestry. Those were what we called homologous traits, and our example last semester was comparing the bone structure between different types of mammals. You can see drastically different types of mammals, whether it's a human, a cat, a whale, or a bat. They've got the same bone structure. One upper arm bone, two forearm bones, a collection of wrist bones and hand and finger bones, all in the same locations. The only difference is the proportions of those bones as they have adapted to life on the plains, life in the water, or life in the air. So similar bone structure adapting in different ways due to different habitats is homologous traits. These do tell us something about evolutionary relationships. I mentioned these in our discussion of the Cenozoic time period in our last discussion because it showed, we saw these slides at least, that during the Cenozoic time period you had these rapid bursts of mammal evolution. And the ancestral mammal that derived from one of those stem reptiles had 5 fingers and 5 toes. This was the ancestral form of mammals. I believe I had shown, I'm just hoping you remember this from last time. I pulled up some kind of picture that had a bunch of mammal skulls. Do you remember that? And it showed all the mammal differentiation, how there was a basal mammal that had 44 teeth, and then as mammals specialized, some of them lost teeth, or they specialized for eating grains or eating meat, whatever it may be. Some mammals are going to retain the primitive 5 fingers and 5 toes like we do, but other animals are going to start to evolve these highly derived forms that aren't going to look anything like the ancestral form. So this is where we get to introduce these terms ancestral versus highly derived. So ancestral, derived. You can see that on the way of mammal evolution, as mammals were trying to cover greater distances, we said their bone structures kind of lengthened out, and they also had a reduced number of digits. So we see a reduction from 5 digits to 4 digits that you would find in cats, like cheetahs. You can see a further reduction of 2 main digits and 2 highly reduced digits in the back. This is what you would find on pigs and cows and deer. And then you can see the single digit, the most reduced form, is what you would find on horses. They're walking around on just a single digit. Anyway, this no longer resembles the primitive ancestral form. This is a highly derived form. This is now using a blue dot to show this node. So the term nodes are going to refer to these branch points, which I also sometimes call common ancestors. They're all the same things. This branch point represents a common ancestor. It's sometimes just referred to as a node. So we've gotten that. When I draw these things on the board, I typically will draw them like this rather than the sideways version that you see on the slides. But if we can just adjust our minds to this for a second, the node would obviously be circled in red. You can see that that represents a common ancestor, and each branch is going to take us to the taxa that would be shared by this common ancestor. Maybe another note is that you can see these names are now below these branch points. Those names are not the names of the common ancestor. Those are the clade names. And just to show that these are clade names. We had seen clade names previously hovering out here on the ends of the taxa. We said there are smaller clades nested inside larger clades. So this clade of Amniotes is a common ancestor that has an amniotic egg, and all of its descendants. That smaller clade is nested inside of this larger clade of tetrapods. This term sister taxa is going to refer to taxa that share a recent common ancestor. We'll look at this node or this branch point circled in red. This represents the common ancestor between the Saurischian dinosaurs and the birds. These are more closely related to one another than either one would be to any of the other taxa on the tree. So we would refer to these two as sister taxa to one another. Here, if I can change our point of focus, if instead of focusing on this branch point, again, the common ancestor tells you the sister taxa. They're going to be shared by any taxa that are shared by this one common ancestor. So in this zoomed in version, our sister taxa are just the birds and Saurischian dinosaurs. But if I zoom that out to this second branch point here, and we think about what are the sister taxa of that common ancestor, well, we go one direction, takes us to the Ornithischian dinosaurs, and the other branch takes us to both the Saurischian dinosaurs and the birds. Again, my circles kind of overlapped there. I couldn't prevent that. But think of the sister taxa to this common ancestor as one being Ornithischian dinosaurs and the other branch taking you to these two taxa, Saurischian dinosaurs and birds. So just pointing out that sister taxa. Let's try it with a new one. This one says, what are the sister taxa of the mammoth? And when we read this chart, we can see the mammoth over here. If we go back there, right there is our node. So sister taxa are going to be anything on the opposite branch here. We know one sister, one branch is the mammoth, so the other branch is going to contain the other sister taxa, and that takes us to three taxa. There are the Asian and African elephants. So all three of these would be the sister taxa to the mammoth. You find that node, you find that branch point, and that shows you the two branches in which the sister taxa are found. The next one says, what are the sister taxa of the African elephants? So this is kind of grouping together African elephants as one, and it's asking what are the sister taxa to those two African elephants. So that's showing us that this is one of the sister branches. This would be the node. And what's on the other branch there are the Asian elephants. So Asian elephants would be the sister taxa to the African elephants. Okay, back to seeing cladograms drawn this way. If we wanted to list the sister taxa of the dinosaur clade from this view, again, there's our node or the branch point. So we would follow this branch out to Ornithischian dinosaurs, and we would follow the other branch out. In this case, this branch has taken us to two taxa. It's taken us to those non-avian dinosaurs and the birds. Okay. Here's a cladogram that doesn't mention anything about geologic time scale, and you can kind of tell that immediately by looking at it because these branch points are nice and symmetrical, and they happen, almost at similar times, and that's just not how it works in nature. So cladograms can sometimes be drawn to make it easy to write all the names down. But in this case, it doesn't mention anything about geologic time scale. Here's one that does mention geologic time scale, and you can see how these branch points are not always at a consistent interval. But you can see these, like we mentioned before, these rapid bursts of evolution that happen right after things get shaken up. This would be more along the lines of that punctuated equilibrium model that we talked about. So here's one again that we saw earlier. It has geologic time in there, and that's going to allow us to answer some additional questions like what is the approximate time that the Asian elephants diverged from the African elephants. If we look on here, Asian elephants diverged from African elephants. There's that branch point, the node that we talked about. It is somewhere between 2 and 5 million years ago, so anywhere between 3 and 4 million years ago are when those Asian elephants separated from African elephants. They've been on their separate directions ever since then. We shouldn't ever expect, I'm going to pull that thing out of the wall before too long. We shouldn't ever expect those Asian elephants to evolve into African elephants just because they're the most closely related species to one another. That would be like my misconception that I mentioned earlier when I was thinking just because chimpanzees and humans are the most closely related living species, we shouldn't expect chimpanzees to evolve into humans. We've been on separate branches for millions of years. Just like the Asian and African elephants have been on their own branches for millions of years. You can see that the two branches continue to diverge. So that branch of African elephants has continued to diverge into two separate species. Okay, we answered that question. The next slide is no longer showing geologic time scale, and we've got a question that says, can you determine the approximate time when mammals diverged from reptiles? And from here you cannot, because there's no mention of geologic time scale. But the second question you can answer. It just simply says, can you determine whether or not mammals or birds evolved first? And sure enough, mammals evolved well before birds. Mammals show up in the fossil record before feathers show up. So mammals came before birds. One other thing to notice, I don't think I've been pointing it out too much, but these purple lines that you see that are right across these common ancestors, those are the acquisition of traits. And the characteristic or the trait is typically labeled like the presence of feathers. When feathers shows up, this leads to typically a whole new group of organisms, in this case birds. I'd mentioned characteristics can be lost as well as gained, and just because organisms lose a characteristic doesn't mean they're no longer part of that group. So I said snakes have lost their legs, but they're still tetrapods. They're still part of the tetrapod group because their ancestors had legs. The fact that they lost their legs is a derived form. It's an adapted form. So, we need to be able to ask the question, what was the primitive form and what is the derived form? That takes us into these terms. Shared primitive characteristics, which their name is pretty descriptive. These are characteristics that are going to be shared by every member in the group. So they've been around, they're very primitive characteristics, so everybody in the group has them. Shared derived characteristics, meaning that they're shared between some members of the group, but not all members have it. They're derived. It's a specialized version, shared between some, but not all. And then the last one, unique derived characteristics, are easy because these are found in only one taxa under consideration, unique to a single group. Let's only consider the ones that are in this green box. So that means we're just thinking about the tetrapods. From the perspective of tetrapods, that characteristic of having 4 limbs you can see is shared by every member of this group. Amphibians, mammals, reptiles, birds, they all are tetrapods. So that's a shared primitive characteristic. Let's now, instead of looking at the tetrapod limbs, let's switch to looking at the shelled egg. That shelled egg, from the perspective of tetrapods, not all tetrapods have a shelled egg. Some have it, but amphibians don't. Some, but not all, have that. This is a shared derived characteristic. I want to point out here that whether we're talking about shared primitive or shared derived characteristics, it's a matter of perspective. So let's put that same slide back up. There's our original green box. Remember, from the green box perspective, if all we care about are those organisms in the green box, then having 4 limbs is shared primitive. Everybody has 4 limbs inside the tetrapod box. But just for a second, if we can broaden our perspective to include the blue, anything inside the blue box. If we include lungfish, they don't have 4 limbs. So all of a sudden, tetrapod limbs goes from being shared primitive to now being shared derived. Tetrapods have it, they're sharing the limbs, but lungfish don't have it. So a characteristic that some members have, but not all members of a group, we refer to that as shared derived. Just zooming in from the perspective of birds, if we just isolate our perspective to birds, feathers then could be shared primitive because all members of the group have feathers. So again, it's kind of a matter of perspective. Let's practice. Actually, we got one more and then we have some practice. This is another one of these cladograms that is showing us a good example of unique derived, at least one unique derived character. That would be the hair that's found on mammals. So only one taxa in consideration has this particular characteristic. Here are the practice questions. If we're not careful, they can seem repetitive, but let's see if we can tackle each one. The first one is, which character on the cladogram is a unique derived character? And the characters, again, are anything here in purple. So we have the characteristic of having 4 limbs. We have the characteristic of producing a shelled egg, or we have the characteristic of producing milk. Which one of those characteristics is unique derived? Yep, that's easy. That's the milk. Only one taxa in consideration has that unique derived characteristic. But let's look at the second one. Which characteristic on the cladogram is shared primitive for tetrapods? So our perspective is looking at the entire tetrapod group, and we're looking for a characteristic that all tetrapods share: legs. So now legs are our shared primitive characteristic. Hold on to that. Let's now move to the third question. Which character on the cladogram is shared derived for tetrapods? So now we're looking at the tetrapod group, and we're looking for a character that some have, but not all tetrapods have. Amniotic egg. All of that makes sense. But now let's change perspectives. This last question wants us to change perspectives and look at the amniotes. So forgetting about the tetrapods and just looking at the amniote clade, which one of those characteristics is shared primitive for amniotes? The egg, the same egg that we just said was shared derived when we're looking at tetrapod perspective, is now shared primitive when we're looking at the amniote perspective. Don't let that confuse you. You may see some questions like that on the quiz or the exam. But that's the last of the slides. I can back these things out a little bit if I stirred up any questions in there. There they are. To recap, we got exposed to a lot of these cladograms, this drawing of evolutionary trees, and we want to draw monophyletic groups, what we called clades, and we want to avoid paraphyletic groups or polyphyletic groups that don't accurately show evolutionary relationships. Are there any questions at all? Okay, then I'll end this, maybe.